Hi, this is Dr. Lonville, and this is the pre-lab for Lab 4, which is running an Agarose gel. What we're going to do now is prepare, run, take a picture of, visualize, and interpret an Agarose gel. Agarose gel electrophoresis is a very standard technique in molecular lab, and one that you can get good at and one that is not too difficult. So, as opposed to protein gels, DNA and RNA gels are run on agarose, which is a carbohydrate. And here's the stuff right here, agarose. It's not the same as agar like you use to pour the plates that we grow the worms in. This is agarose. This is a more purified form, and it's more expensive. So this little jug here is probably about $300. What we're going to do is weigh out a given amount of agarose according to the size of DNA fragments and RNA fragments that we want to separate. So by increasing the amount of agarose as a percentage of the total liquid in the gel, you can decrease the pore size that is generated when the gel polymerizes. So a 1% gel is what we're going to make today, and that means that the dry agarose we weigh out is 1% of the total weight. So the volume of the gel we're going to pour is 100 milliliters in this example, and 1% of 100 milliliters is 1 gram, assuming that distilled water weighs 1 gram per milliliter, and that's what we're assuming. So I'm going to go over here to the scale and weigh out 1 gram. If I wanted to separate DNA particles that were smaller than that, I would weigh out 1.5 grams or 1.5% gel. I can go up to, say, a 3 or 4% gel for really small holes in the gel, which is good for separating small things. Bigger things, bigger fragments of DNA, you want a lower percentage of the gel, which is bigger holes in the gel. Smaller pieces of DNA, you want smaller holes in the gel, which is a higher percentage of gel. And then I'm going to weigh out 1 gram of agarose. This is what the agarose looks like. Okay, that's a little more than a gram, but it'll work. Cover this back up. Then I'm going to put my 1 gram in an Erlenmeyer flask. 200 milliliter Erlenmeyer flask is fine. Just put the dry powder in there, and then I'm going to add tris acetate EDTA. You can use tris acetate EDTA or tris borate EDTA buffer. We're going to use tris acetate EDTA, or TAE for short, for this one. And I'm just going to use the gradations on the outside of the Erlenmeyer because this is not super precise what we're doing. You could use a graduated cylinder. No real need to for what we're doing today. Okay, so 100 milliliters of total solution, and you can see that it's very cloudy in there. Now what I'm going to do is I'm going to heat up this solution in a microwave. I'm going to do it gradually until I see a completely clear solution. Okay, here we are at the microwave. This is a microwave, of course, that is never used for food, and you can see it's pretty funky in there. Maybe your microwave looks like this. But we're just going to put the gel, the agarose in there with the Erlenmeyer, and we're going to start it up for about 30 seconds initially. Okay, so initially you don't want to boil completely because with all that dry agarose in there, it can boil over. So I like to go for about 30 seconds initially, stop it, give it a swirl, and then go back in about 10 or 20 seconds at a time. Okay, we're going to check on the progress now, and you can see hopefully that it's much more clear than it was, but there's still particles floating around in that agarose. You want to do it until you can't see any particles floating around, that it's completely clear. You definitely want to be here the whole time that you're microwaving, because if you walk away, you'll end up with a big mess. And you want to use these to handle the Erlenmeyer because it gets really hot. You can see probably in there it's starting to boil, so I'll pull that out. And I will swirl it. And probably hard for you to see on there, but there still are particles swirling around. So I'm going to give it a couple more tries here until it looks completely clear. All right, now I think we're good. And what I'm going to do is I'm going to go over here to the sink, and I'm going to rinse off the bottom of this. Make sure you get none of that tap water actually in your flask. But this, all I'm doing now is cooling off the agarose. You don't want it to get too cool because it'll solidify, but just coming out of the microwave, it's a little bit too hot to put into your gel. So you should be able to hold on the bottom of your hand without it being uncomfortable. And if you can, that's about 50 degrees C, and that's about right. Okay, ideally you would have this prepared ahead of time. You want to put your mold in the gel box. This is the tray that your DNA gel is going to be poured into, and it forms a mold for this. You're going to put it so the gaskets are facing the long side of the gel. And what I did is I just put some TAE buffer in the gel and got those gaskets a little bit wet so that I can press them down and form a good seal. If you don't do that, the gaskets tend to roll up on you and you won't get a good seal. To get a good seal, it should look like that, in that the gasket is slightly flattened and you can see it through the plexiglass. If it's rolled up on a corner or something like that, it won't hold water. Then the other thing you want is to put a comb in place. This is a 10-place comb on one side and a 10-place comb on the other side. I'm just going to put that in a slot on the top so that I can have my samples at the top there. Then finally, I'm going to add some dye to the gel. This is dye that binds to DNA. Okay, see that dye there? It binds to DNA, and so as the DNA travels through the gel, it'll be labeled with this dye, and then when we put it under fluorescent light, you'll be able to see the DNA bands. Depending on the lab that you're in, I'm just going to swirl this around to make sure that it's nice and even. Okay, depending on the lab you're in, they either put the stain in the gel like I'm doing right here, so that's nice and even, or they run the gel without stain and then stain afterwards. So they put the whole gel after it runs into a solution of stain. I don't like doing that for a couple reasons. One is you use a lot of stain and you have a big Tupperware full of stain. And the other is that while you're staining after the gel, it's not under electrophoresis condition, so it's not under voltage, and so your bands can move, they can diffuse, and you get not as sharp a gel. So right after you are done pouring, like I just did there, just poured it in there, you need to rinse this out immediately at the sink. So I'm gonna go do that. Okay, so this has been sitting for about 10-15 minutes, and you can see that the gel is much more milky looking. So now I'm going to, and if I move this, it's solid, okay? So now I'm gonna remove the gel, and this is the part where you really need to grip it hard so that it does not go flying across the room. And there you can see the comb inside the gel there. I'm going to rotate my box, oops, sorry, I had it right the first time, so that the red pole is on the bottom of the gel. So the top and bottom, top is where your samples are. The samples run to the bottom. Always remember the phrase, run to the red. So nucleic acids are negatively charged. They are running toward the positive pole, okay? And so that's the anode because it attracts anions. This is the cathode, the negative pole, because it attracts cations, which are positive ions. A little bit confusing there, okay? So now what I'll do is I'll pour the TAE buffer over on top of the gel, okay? And it should just barely cover the top of the gel, okay? You don't have to fill it right up to the tippy top. Then I'm going to very carefully remove the comb here. That. So now what I'm going to do is I'm going to load my gel. The first thing I'm going to load is these DNA plus size standards, the DNA ladder. 5 microliters should do it for this. So I'm going to load 5. You want to get your tip right on the edge of a well and then watch it fill up from the side. You don't want to put your tip into the well, but just on the edge of the well. So it's just resting on the edge, like I'm resting on the edge of this gel box. So not in there, because I can poke a hole in the bottom of the gel and then your sample will go out. All these samples have glycerol in them, which makes them really viscous and heavier, more viscous than the running buffer. And so even though there's running buffer in those gels, when I put it in there, it'll fall to the bottom of the gel, okay? And I have my shoulders nice and braced. I can look right on top of where I'm going, and I can add my sample there, and that is in that well, okay? I'll try to show you, once I get all these loaded, I'll try to show you what they look like from the side so you can get an idea. PCR sample, I'm stealing this from somebody in the lab, and so if you're missing a PCR sample, it's probably my fault. It already has dye in it, that's that green stuff, and it's got the glycerol in it, so you can just load that directly on there. And it should be 20 microliters, I think, was the size of your reaction. I'm going to pipette this. Hopefully you can see it. Okay. I'm going to pipette this right next to my other sample. The wells can hold as much as 50 microliters, so putting 20 microliters in there should be no problem. And what you want to do is you want to pipette nice and slowly and let your sample fall to the bottom of the well so that it doesn't contaminate the well next to it. That's really important for these PCR sorts of gels, because what you're doing is you're trying to find out the difference between negative control and positive control. So negative control should be blank, shouldn't have any reaction in there. Positive control should have a reaction in there. If you pipette too fast and some of your positive control leaks over into your negative control lane, you'll get a band in both. And you don't know if that's what really happened or is it because you loaded the gel too fast. So take some time doing this. You're going to do it with other people in the group. You don't have forever to do it because the samples will start to diffuse together, but you got like 10 minutes. So have everybody ready to go. Everybody should take a shot at loading the gel, and if you mess up, you mess up. But the whole idea is to get them on there and get the electrophoresis started so that now for your RNA is not labeled. It doesn't have any dye in it. And so hopefully you can see on this parafilm, that's what I was trying to say. My old guy brain wouldn't work there. I'm just taking 5 microliters of the no dye RNA and I'm putting it on the parafilm, okay? And then I'm going to add 1 microliter of, it's called 6X loading dye. So 6X is 1/6 of the total of your sample, so I'm going to have 1 microliter of that dye to the 5 microliters that is on that parafilm, and then I'll mix it up right on that parafilm. So I'm going to add my 1 microliter right to this sample, and then I will mix them together right on the surface here. Suck up that sample of the RNA, and I'm going to put it in the lane right next to where I put my PCR sample. Okay, and that's my sample of RNA. Okay, so those are loaded in there now. Now what I'm going to do is I'm going to start the electrophoresis. It's important that you cover the gel because you don't want 150 volts going through you. Sometimes these electrodes don't fit on there very nice if you put the cover on, so I'm just going to tilt the cover a little bit so I still get the protection, but I can put on these electrodes. Red to red, black to black. Okay, you're going to have the same red to red, black to black on the power supply. The power supply you want at 150 volts. Approximately 150 volts, and then what you should see right away is you should see bubbles, okay? And then you want to pay attention to the migration of your samples, and they should be moving into the gel rather than moving out of the wells. So if you hook up your electrodes the wrong way, red to black and black to red, they will move out of the gel instead of into the gel. And you can already see, hopefully I can see anyway, you can start to see those samples and the color moving into the gel. We'll check on this periodically. It takes about, in real time, it takes about 45 minutes to run. Okay, hopefully there's a little bit of a glare there. But I put the gel against a white box. This is about 5 minutes into the run. You can hopefully see that the samples have moved out of the wells, and you see in the second lane there, you see a yellow band that migrates. That's a dye that migrates at about 50 base pairs. And then in the last lane there with the RNA, it's got a blue band, and we'll check and see how they separate as the gel progresses. Okay, it's been about 45 minutes. Hopefully you can see a yellow. Okay, so what I have here is the gel. Notice how I'm holding that, and I've got my thumb on one side and my fingers on another side. The gel is really slippery, and if I don't do that, it'll slide right off. Now I'm going to place that on a UV tray, which is something specific to this instrument that will take an image of the gel. And I'm going to put the tray into the machine. Okay, and close. Okay, so I'm going to say user, image lab right over here. This is the program I want, so I'm going to double click on that. Okay, I'm going to have a new protocol. It's gel imaging. I'm going to select, all right, I'm going to select nucleic acid stains and then gel red as my stain. I'm going to say run protocol. Okay, and then here's the image of the stain. Okay, so what does this mean? This is a 100 base pair ladder right here. Okay, so I'm going to say 1, 2, 3, 4, 500 base pairs. Again, 1, 2, 3, 4, 5. 5 is doubly bright. And this product I've got here, I ran a positive control, is between 100 and 200 base pairs, so that makes sense. It's a little smeary. I could run this gel out longer. There's plenty of gel left and it might tighten up, but I see a band right there. And then these are your RNA. This is your RNA sample, and you can see it's a big smear. There's a little bit of a hint of a band here and here. The big smear means that this RNA is starting to degrade. As it degrades, it has many size pieces in there, and they show up as a smear on the gel. What you're looking for high quality RNA is 2 very crisp bands on that RNA. So that's how I would interpret this gel. I can save this gel. I can take a picture of it with my phone and put it in my notebook. So this is a positive control. I would say this worked. I would run this next to a negative control. I would expect nothing. Sometimes on negative controls, you see a very small band below the 100, and those are primer dimers. Those are just from the primers, but you won't see this band here. So this is a positive control that worked. Okay, when I'm done with the gel, I'm just going to put it in chem waste, just like this. And then what I'll do is I'll rinse off this tray with distilled water and put it over there by the machine.